Welcome back to the Deep Dive. Today, we're not just skimming headlines, we're diving deep into some pretty specialized source material.
That's right. Think reports, intelligence summaries, policy docs, stuff that usually stays behind closed doors.
And we're distilling it down. Think of this as a specific intelligence briefing tailored for, well, for you listening right now.
Exactly. This one's really for the pros navigating the cutting edge of biotech today. So immunologists, vaccinologists, microbiologists, bioengineers.
You're the ones working with dual-use tech every day.
Right. And the material we've got lays out the strategic context, the funding landscape, the policy pressures surrounding your research.
Our mission here is to give you that analyst's view. We're looking hard at U.S. government funding tied to biological weapons and, importantly, biodefense.
We're focusing on the technical side, the policy side, and how those two things intersect, sometimes awkwardly.
It's a complex picture. There are legacy decisions from decades ago, some, frankly, disastrous proliferation issues involving U.S. funds.
Oh, yeah. We'll definitely get into that.
And then the really high-stakes ethical tightrope of current dual-use research.
And the sources really force us to draw some fine lines. Distinctions between the old offensive programs, what's needed for modern defense.
And critically, these really shocking allegations about fund diversion. How money meant to stop proliferation might have fueled the very thing it aimed to prevent, in the 90s specifically.
That history directly shapes the scrutiny your work gets today. It really does.
But here's the hook, the big takeaway for our technical audience today. Policy decisions, past and present.
Like shutting down programs or funding new ones.
They directly impact your ability to develop medical defenses, vaccines, treatments, diagnostics. It's all connected.
And maybe the biggest lesson from the sources, how a lack of oversight, especially in international science collaboration, nearly led to disaster, funding the threats we were trying to eliminate.
We need to trace that. How did a field born out of necessity get tangled up in these strategic political failures?
Okay, so to really get the current biodefense situation, you have to look back. Where did it start, and how did it, well, abruptly change?
The U.S. had a formal offensive biological weapons program, BW program.
Right, for 27 years.
Yeah.
Kicked off in 1942, ran all the way to 1969. That period set the benchmark, really, the scientific baseline for what a state-level offensive capability looked like.
And that program ended, formally. President Nixon, 1969, executive order.
Crucial point. And the U.S. followed through, destroyed the stockpiles. Diplomatically, that was huge. Paved the way for the Biological Weapons Convention, the BWC, in '72.
A major step for arms control.
Absolutely. A policy victory, no question. But,
Wow.
and this is clear in the sources, that policy decision came with a cost. A serious scientific and operational cost.
How so? For medical readiness.
Directly. It hit military, medical, public health preparedness pretty much immediately. For our audience, those developing countermeasures, vaccines, diagnostics, drugs, this history explains a lot about today's funding debates.
Okay, let me push back on that a little. Because stopping offensive weapons development sounds like a purely good thing, right? Couldn't the defensive research units just keep the necessary knowledge going? Did they really need the offensive part?
That's the tension, isn't it? The sources are explicit. They say the closure had a serious and limiting effect on developing vital medical defenses. They specifically mention vaccines, antibiotics, other treatments.
So the limitation wasn't ethical, it was practical, technical.
Technical, exactly. Defensive work, to be truly effective, has to understand the maximum threat, the worst-case scenario.
Okay, maximum threat envelope. What does that mean in technical terms? What parameters are we talking about that only offensive research would really push?
We're talking about optimizing for weaponization, for large-scale dissemination. Look, a microbiologist can grow a pathogen in the lab, right? Keep it stable.
Sure, standard practice.
But that lab strain is unlikely to have the sheer robustness of a weaponized version. Offensive programs perfected things like high concentration yields. They maximize stability in aerosols for delivery.
So it survives being sprayed out.
Right. And resistance to standard decontamination methods you'd use in the field. These aren't trivial things.
So you're saying an adversary isn't just going to release some bug they found, they'd release an engineered version, designed to survive the bomb, the air, and overwhelm the initial immune response.
Precisely. So if you're a vaccinologist and you design your vaccine based on a standard clinical isolate, a natural strain,
Okay.
it might just fail against a highly optimized, high-yield offensive agent, one engineered for maximum stability when aerosolized. Those offensive programs, they set the technical benchmark for lethality and transmissibility.
So when Nixon shut down the research that defined that state-of-the-art weapon,
The U.S. medical defense apparatus suddenly lacked an up-to-date stress test. They lost the high water mark.
The knowledge gap wasn't just which pathogens were dangerous, but how an intelligent adversary had optimized them to get past our defenses.
Yes.
Exactly. That meant the defensive programs, the countermeasure work, they lacked the proper virulence standards. How do you measure if your vaccine is good enough?
If you don't know the stability of the best aerosol version.
Right. You can't reliably test your air filters. You can't be sure your vaccine gives enough protection against the maximum possible dose delivered via the most effective route. This historical limitation, that's why today's biodefense efforts, which we'll get to, are forced into things like replicating specific optimized strains or creating these complex virtual weapons.
They're trying to rebuild that comprehensive threat picture, the one that was effectively dismantled in 1969.
That's a good way to put it, rebuilding the threat picture.
Okay, so that initial tension, policy goals versus operational needs, it really sets the stage for something far more dangerous, the proliferation risk after the Soviet Union collapsed.
Right. A huge strategic challenge. And the U.S. response was the Cooperative Threat Reduction Programs, CTR.
Designed to manage the fallout, basically. The intention was absolutely right. Noble, even necessary. The USSR collapses. Suddenly you have thousands of top-tier biological weapon scientists.
Highly skilled people, potentially unemployed.
Exactly, with immense expertise, potentially unsecured deadly materials. CTR was designed to stop proliferation, prevent those scientists from selling their skills, or worse, their stocks, to rogue states or terrorists.
So the U.S. government poured money into this. State Department, Defense, Energy, all involved.
A lot of money. Trying to redirect these scientists towards peaceful research.
Paying brilliant minds to pivot from, well, death to diagnostics, a critical intervention. But the structure of the Soviet BW program itself created a massive loophole.
A loophole named Biopreparat.
Ah, yes. The hidden elephant, as you called it earlier.
It really was. And it's the ultimate example of an intelligence failure impacting policy. For our audience to understand, Biopreparat wasn't just a few labs. This was the engine of the Soviet offensive BW capability. Massive. Clandestine.
And the key, it operated under a civilian cover.
Completely. Posing as a network for public health and vaccine research.
The perfect biological Trojan horse. Seemed benign, doing civilian work.
All while secretly violating the 1972 BWC. They kept doing offensive BW work well into the early 1990s, maybe even later.
So when U.S. non-proliferation money started flowing into post-Soviet science,
Biopreparat just adapted, positioned itself to capture that funding.
which leads straight to these really shocking allegations of fund diversion, a systemic intelligence failure right there in the sources.
Russian scientists themselves eventually claimed it. U.S. money, meant strictly for civilian science for non-proliferation, was secretly funneled to Biopreparat.
How did that even work? How did they pull it off?
Well, Biopreparat reportedly reborn itself, became a state-owned drug company on paper.
Ah, so it looked like a legitimate recipient for conversion funds for research grants.
Exactly. A bureaucratic shell game, exploiting the very agencies trying to help secure things.
And it was led from the top, wasn't it? By a key figure.
General Yuri T. Kulinin, not just some manager, he was the Biopreparat director and a former intelligence officer. He'd spearheaded the Soviet BW program. This guy knew deception.
Strategic deception.
Absolutely. From the top down.
And this is where it gets almost unbelievable. The sources allege Kulinin diverted funds from NASA grants.
Yes, NASA grants. At least 10% of several grants intended for space biological research allegedly got shifted to his old Biopreparat infrastructure.
Wait, space biological research, what does that even mean in this context?
Think about things like studying microbial effects in zero gravity, how microbes behave on spacecraft, that kind of thing.
So NASA money meant for studying microbes in space.
With allegedly helping prop up the remnants of the clandestine Soviet biological weapons program. The disconnect is just staggering.
It points to a massive oversight problem. U.S. side and NASA involved here. How could major U.S. agencies be so unaware of who or what they were funding?
It looks like a breakdown in communication. Institutional memory failure. U.S. officials later admitted, according to the sources, they just didn't know Biopreparat's classified history.
They didn't check.
They acknowledged they should have checked with national security officials before okaying aid to a joint venture involving this group. They treated a major BW entity like just another startup pharma company.
And this was happening under the Clinton administration.
Yes, and high-level officials admitted later they were less than meticulous in forcing the ban on aid to Biopreparat in the mid-90s. It was a known risk, but enforcement was lax.
The sources mention some serious anger about this internally.
Extremely serious. One senior national security official was reportedly furious, said USAID and NASA were essentially running their own foreign policy, undermining non-proliferation goals through, frankly, a lack of due diligence.
Was intelligence just not sharing the threat info?
That seems to be part of the systemic problem, yes. The sources suggest that early on, intelligence officials were reluctant to share the critical details with the civilian agencies handling the money.
So you have national security holding back key context.
And civilian agencies moving forward, basically blind, with huge sums of money. It created the perfect storm for Kalina's deception.
Okay, the bureaucratic and financial mess is bad enough, but the technical red flags were even worse.
Truly horrifying, and this should really hit home for our audience. Microbiologists, bioengineers, this shows the kind of advanced work U.S. taxpayer money might have been supporting.
The sources say internal warnings about this diversion were suppressed.
Reportedly, yes. Suppressed by the Clinton-Gore administration. The warning came from within the U.S. government itself.
From where?
A Centers for Disease Control and Prevention representative, CDC, expressed extreme concern about a specific project.
Funded how?
Through the State Department-funded International Science and Technology Center, the ISTC, another channel for this non-proliferation money.
And the CDC, focused purely on public health, saw the danger immediately.
Recognized the lethal potential, yes.
So what was this project? What were they actually working on that raised such extreme concern?
They were working with a genetically mutated measles pathogen.
Okay, measles. Highly contagious.
Yes, and the objective was engineered to exhibit, and this is a direct quote, fast-acting AIDS-like characteristics.
Fast-acting AIDS-like characteristics. In measles, that sounds devastating.
Chilling for any vaccinologist, any immunologist.
Okay, let's break that down for the microbiologists listening. What biological mechanism are we talking about? Fast-acting AIDS-like doesn't mean replicating HIV's long timeline.
No, absolutely not. Measles itself is already an immunosuppressor, right? It temporarily weakens the immune system.
Right, transient immunosuppression.
To get fast-acting AIDS-like effects, you'd be aiming for acute catastrophic immune collapse very quickly. This likely means engineering the measles virus, an RNA virus,
Okay.
to express specific proteins. Proteins designed to target and wipe out critical immune cells, like CD4 plus T helper cells, much faster than normal measles. Or maybe engineering it to trigger a massive, uncontrolled cytokine storm, something that just overwhelms the body's defenses in days or weeks, not years.
So using the measles virus, which spreads easily,
Exactly. Highly transmissible platform.
to deliver a payload that causes rapid acquired immune deficiency. It essentially turns a common disease into a hyper-optimized, dual-purpose bioweapon.
Letting secondary infections, normally containable ones, become immediately lethal.
That is deeply disturbing.
The CDC representative reportedly warned there'd be a massive outcry if the public knew U.S. tax money was funding work like this, creating such an advanced, high-risk threat.
It's the nightmare scenario of non-proliferation funding, isn't it? Money meant to reduce threats.
Directly risking the creation of next-generation biological weapons,
Yeah.
all under the guise of civilian research conversion.
You end up funding the ghost of the very program you're trying to dismantle. And that ghost is working on cutting-edge genetic manipulation.
A profound and frankly terrifying lesson in what happens when oversight fails, especially at that boundary between civilian science and military goals. So those historical failures, particularly the Biopreparat mess, definitely led to a shift, a much more rigorous, sometimes controversial approach to biodefense domestically.
Which brings us to the present challenge, dominated entirely by this concept of the dual-use spectrum.
Right, and the constant need to anticipate the kind of high-end threats that, frankly, we might have lost track of or even inadvertently funded back in the 90s.
And for our audience, the microbiologists, immunologists, bioengineers actually doing the work, dual use, isn't some abstract policy term.
No, it's your daily reality. It's the central operational dilemma.
Every breakthrough you make, maybe in vaccine tech, gene sequencing, diagnostics, intended for good,
But
public health.
can potentially be diverted.
Yeah.
Or used as a blueprint for a weapon. It's inherent in the science.
So current U.S. funding, heavily weighted towards managing this exact gray area, a lot of it flowing through the Department of Defense, DoD, and the intelligence community.
And their operations really highlight what we might call the offensive-defense paradox. Meaning, to build effective defenses, vaccines that work against the worst threats, better detectors, treatments you first have to understand or even simulate, the highest possible threat level.
Which means doing research that can look awfully close to offensive work itself.
Exactly. It walks a very fine line.
Can you give us a concrete example, something high profile, where DoD pushed that technical boundary for defensive reasons?
Sure. Back in early 2001, the Pentagon had plans. They were going to genetically engineer their own modified anthrax strain.
Their own modified anthrax, not just working with existing strains.
No. And this wasn't just some standard lab variant. The sources are clear. The purpose was explicitly biodefense, to duplicate.
Duplicate what?
What they believed was a more potent Russian strain, something potentially more dangerous than what was publicly known. They needed to test their defenses against that specific perceived threat.
Okay, for a microbiologist listening, what does more potent likely mean for Bacillus anthracis? What kind of modifications would necessitate actually replicating the threat, not just studying known sequences?
You're probably looking at things crucial for weapon delivery and effect. Maybe enhanced spore stability in an aerosol, right?
Sure.
How long does it hang in the air? How far does it travel?
So dispersal characteristics?
Yes. Or maybe modifying the toxins themselves, the kinetics of how the lethal factor and edema factor work, making them act faster, perhaps, or making them resistant to standard antibiotics like Cipro.
Things that make it harder to treat, deadlier upon exposure.
Exactly. If the adversary's strain is optimized for those things, your vaccine, your therapeutic, it has to be tested against that specific optimized threat agent, not just a reference strain from a culture collection.
So they felt they had to replicate the threat to develop a reliable countermeasure.
Okay.
And since physically creating and testing large amounts of such agents is problematic.
Highly problematic under the BWC, yes. Which is why they also rely heavily on virtual weaponization.
Ah, the modeling approach.
Exactly. This defensive research leans heavily on classified literature, on intelligence reports about adversary capabilities, and sophisticated computer modeling.
To create what? Virtual pathogens?
Yes. And virtual weapons systems. This often goes through specialized contractors. Battelle is frequently mentioned in the sources, experts in simulating these complex biological and physical systems.
Okay, for the bioengineers listening, what does virtual pathogen actually mean operationally? Is it just analyzing the genetic sequence or something more?
Oh, it's much deeper than just sequence analysis.
Yeah.
Think comprehensive computational fluid dynamics CFD, modeling how an aerosol cloud of this agent would disperse in a city, in a subway, over open terrain.
Wind patterns, humidity, UV degradation, all factored in.
All of it. Modeling stability, drift. Then modeling infection kinetics in a population. How fast would it spread? What mutations might arise? Predicting resistance profiles against our current drugs.
So it's predictive epidemiology on steroids, linked to physics.
Pretty much. A virtual pathogen lets the military run massive what-if scenarios, predict casualties, test response strategies, identify weaknesses in infrastructure, all without actually releasing kilograms of deadly agent. It's simulation for strategic planning.
But crucially, you can only go so far testing a physical defense, like a new gas mask filter or a vaccine against a purely virtual threat.
Precisely. Which explains why the physical threat replication, like that modified anthrax plan, still gets proposed or carried out in contained ways.
Yeah.
Virtual models inform strategy, but physical testing validates the actual countermeasure.
Which brings us to the intelligence side. You mentioned the CIA.
Right. Their program called Clear Vision.
What was the focus there? Technical objective.
Clear Vision was secret, CIA funded, focused primarily on the delivery mechanism, the weaponization part.
Not just the bug, but the bomb.
Exactly. They specifically acquired and studied Soviet-designed biological bomblets. The goal was to understand exactly how they worked. Dispersal patterns, payload fusing. How could they be used against the U.S.? Understanding the delivery system is critical for defense.
But didn't that project also get close to creating new threats itself?
According to the sources, yes. It pushed right up to that boundary.
Yeah.
The project reportedly involved genetically manipulating pathogens.
For what purpose?
To identify specific genes. Genes that could make the pathogen more deadly or more resistant to our countermeasures. Purely focused on enhancing lethality or survivability.
So finding the genetic turbo buttons for weaponization.
That's one way to think about it. Identifying sequences that boost toxin production, maybe, or enhance how the pathogen gets into cells.
But that part was stopped.
That specific aspect, the active live agent genetic modification purely to enhance lethality, was reportedly halted due to political concerns.
Ah, so even within the classified world, there was a line.
A recognition, it seems, at a high level, that while defensive simulation is necessary, actively engineering live agents specifically to be deadlier crosses a legal and strategic threshold very quickly. It becomes indistinguishable from offensive work.
So this whole segment really shows modern biodefense isn't passive. It's this active process of offensive simulation.
Using sophisticated microbiology, bioengineering, computation,
Right.
which constantly puts her right under the shadow of arms control treaties like the BWC.
Which brings us squarely back to the legal framework. The Biological and Toxin Weapons Convention, 1972. The BWC.
The big one.
For anyone working in this field, especially if you're doing genetic modification or developing countermeasures, navigating the BWC, it's arguably the biggest policy constraint, right?
It's the constant tightrope walk.
Yeah.
Absolutely. Because defensive research, especially if you're trying to get ahead of potential threats, like developing resistance to known treatments.
Or engineering organisms to test detectors.
Right. It requires manipulating organisms, creating novel countermeasures, and that inherently bumps right up against the BWC's prohibitions.
Let's pin down the specific treaty language that creates this gray area.
Okay, the BWC is very clear on prohibiting the development, production, stockpiling of biological agents for hostile purposes. That's the bright red line. No offensive weapons. But the treaty explicitly permits research for prophylactic, protective, or other peaceful purposes.
Prophylactic, protective, or peaceful.
And that word protective, that's the key. It's the justification, the necessary exception perhaps, that allows biodefense research to even exist legally.
But the moment you go from researching a pathogen to testing a defense against a realistic weapon, a high-fidelity simulation of an attack.
That's where you wade into the legal deep end, testing against something designed to mimic an enemy's actual delivery capability.
And this is where sources say there's real debate among U.S. officials.
Yes, an ongoing legal discussion. Does testing a fully operational weapon, even if your intent is purely defensive, actually violate the treaty?
And by fully operational weapon, we mean the whole package, not just the bug.
Exactly. Replicating the pathogen and the delivery system. Imagine testing, say, a mock biological bomblet's dispersal pattern over a simulated target area to see if your detectors pick it up. Is that allowed under protective purposes?
What are the two sides of that argument, legally speaking?
Well, those who argue it violates the BWC tend to focus on the spirit of the treaty. They'd say designing and testing a functional delivery system, even defensively, is developing an offensive capability. If the system works, it's a weapon, regardless of intent right now.
So it's too close to prohibited activity, like the concerns around Clear Vision's tactical analysis.
Right. The counterargument, likely favored by DoD and operational folks, is driven by necessity.
How so?
They argue you cannot genuinely test defenses without realistic simulation. How do you know if your sensors work against a realistic cloud? How do you optimize deacon procedures in a contaminated zone? How do you validate protective gear under stress? They say it's impossible without high-fidelity, maybe even high-volume simulation of the attack method.
So therefore, it is covered by protective purposes as long as you don't stockpile agents for actual hostile use.
That's the argument. And that ambiguity, that ongoing debate, it's a critical policy gray area. It impacts research contracts, protocols, everything for people in this field.
Okay, so given that legal ambiguity and assuming adversaries are pushing boundaries,
A necessary assumption in defense planning.
the U.S. has to focus on staying ahead technologically, especially against genetic weapon systems.
Absolutely. The focus shifts. It's not just about old threats like anthrax anymore. It's about countering threats designed with modern tools. CRISPR, synthetic biology.
Moving the defensive game from the organism itself to regulating DNA, in a way.
You could say that. Understanding and mitigating threats at the genetic level.
So what specific technical vulnerabilities is DoD looking at for these next-gen threats?
Well, sources point to tackling systemic challenges, like how efficiently could an adversary actually deliver engineered genetic material to affect a large population? That's a huge hurdle.
Delivery is always key.
Right, so research into aerosol stability of, say, viral vectors carrying CRISPR components or maybe new lipid nanoparticle carriers. And also a big focus on testing and evaluation T&E,
Meaning?
ensuring countermeasures, especially genetic ones like therapeutic RNAs or editing inhibitors, work effectively across diverse populations, different genetic backgrounds, different environments. That's a massive bioengineering challenge. Things don't always scale or translate easily.
Which brings us to a really specific, very relevant example for the bioengineers listening. DARPA's Safe Genes program.
Ah, yes. Safe genes. A perfect example of this forward-looking defensive posture.
It sounds like the ultimate defensive engineering goal, doesn't it?
It's incredibly ambitious, it's proactive, highly sophisticated. The aim isn't just to, say, cure disease caused by gene editing, but to actively control, counter, and even reverse the effects of unwanted genome editing.
Okay, let's get technical. How do you reverse unwanted genome editing? If an adversary uses CRISPR-Cas9, say, to insert a harmful gene or make a dangerous mutation, how do you undo that specifically?
You're talking about needing an antidote editor, essentially. Or maybe a kill switch for the original editing machinery. The core challenge, again, is delivery.
Getting the antidote where it needs to go in the body?
Precisely. You'd likely need highly optimized delivery systems, maybe engineered viruses like AAVs or advanced nanoparticles. These would carry their own cargo, perhaps specific guide RNAs and nucleuses designed to find the adversary's edit.
And then what? Cut it out. Inactivate it.
Either excise the harmful introduced sequence, or maybe deliver something that specifically targets and inactivates the Cas9 enzyme the adversary used, stopping further damage. Or even, potentially, an editor that precisely reverts the malicious mutation back to the normal sequence.
The precision required sounds immense. You have to find the specific bad edit, deliver the fix, and critically,
Avoid causing new problems, off-target effects. Hitting the wrong part of the host genome could be just as bad, or worse.
So the engineering challenges are huge. Delivery efficiency, getting it to the right tissues, and minimizing those off-target cuts.
Immense challenges. Safe Genes is right at the absolute cutting edge of genetic regulation and control. It shows the defensive posture has moved way beyond just antibodies or antibiotics. It's aiming to control the editing tools themselves.
Which, of course, further complicates that BWC debate, right? Because the protective measure itself involves large-scale, sophisticated genetic manipulation.
Exactly. It pushes those legal boundaries even further. Now, while DoD and the intelligence agencies are handling that high-end, often classified biodefense simulation and countermeasure work,
The pointy end of the spear, so to speak.
right, we absolutely cannot overlook the biggest engine for dual-use research in the U.S., and the one most directly relevant to the vast majority of our listeners,
And that is?
the National Institutes of Health, NIH.
Ah, the civilian science powerhouse. This is the critical and often less politically fraught piece of the puzzle.
It is. NIH funds a huge amount of infectious disease research. Basic science, applied science. And almost all of it inherently falls into that dual-use category.
Every time a researcher studies how a virus replicates, figures out a new pathway for infection, optimizes a viral vector for gene therapy.
That knowledge, generated for public health, for understanding disease, it simultaneously informs potential bioweapon design and potential biodefense countermeasures. It's unavoidable.
So the day-to-day research, driven by the need to fight disease and just cure scientific curiosity,
Is the bedrock. It's the foundation of the nation's entire biodefense capability, scientifically speaking.
Yeah.
The sheer volume, the technical depth of NIH-funded work, it's arguably the single most important factor in U.S. scientific leadership in this area.
Okay, let's dive into some specific examples from the sources. Vaccine and therapeutic research funded by NIH. Why are these civilian projects so strategically valuable for biodefense?
Well, the sources highlight NIH funding supporting work on MERS-CoV and obviously SARS-CoV-2 vaccine development. Critically important for public health, of course, pandemics.
Absolutely essential.
But the platforms and the techniques developed through that funding, they are absolute game changers for strategic biodefense readiness, too.
Okay, let's start with mRNA vaccines. Obviously huge recently. What's the specific dual-use value from NIH-funded mRNA work for biodefense?
Speed and flexibility, that's the key strategic value. Biodefense demands rapid response capability against a novel outbreak, or worse, an engineered threat.
Something unknown appears.
Exactly. Unlike traditional vaccines growing viruses in eggs or cells, which takes months, right? The mRNA platform lets you plug in a new sequence and scale up production incredibly fast. Weeks, potentially.
So the rational design principles learn, figuring out the best antigen shape, the best code.
That knowledge, funded by NIH for COVID or MERS, can be instantly pivoted, applied to counter a deliberately engineered pathogen or some unknown disease X that pops up. That speed of deployment is maybe the ultimate defense against a fast-moving biological attack.
A massive logistical and time advantage. Okay, what about structure-based design? For coronavirus subunit vaccines, this is deep immunology and vaccinology.
Right. Structure-based design means using techniques like cryo-EM to get an exact atomic level map of the target protein, like the spike protein on coronaviruses,
The blueprint.
the blueprint. Then you can intelligently design modifications like those famous proline substitutions in the spike protein, to make it more stable, easier to manufacture, and crucially, elicit a better, more focused immune response.
And the biodefense value of that precision.
It means less guesswork.
Yeah.
You ensure the vaccine targets the most important, maybe the most conserved, parts of the pathogen. That's vital if an adversary tries to be clever, tries to mutate other parts of the virus to escape immunity. NIH funding supports the structural biologists, the immunologists, doing that high-fidelity design work. It provides the detailed targeting info for defense.
Okay, one more example. Nanobodies, a more specialized area NIH supported. Why are these single-domain antibodies strategically important?
Nanobodies offer some really interesting advantages, especially for biodefense logistics and application, because they're smaller than conventional antibodies.
Derived from llamas or sharks, often.
Often, yes. Their small size makes them incredibly stable. They can often withstand high temperatures, harsh conditions. Think about field deployment. Storing them without needing a complex cold chain, that's huge.
Easier logistics.
Much easier. Plus, they can sometimes be engineered to bind to tricky spots on a pathogen, little crevices that normal, bulkier antibodies can't reach.
So better targeting, potentially.
Potentially. And in a biodefense scenario, this translates to maybe developing next-gen therapeutics or diagnostics that are robust, highly portable, and maybe even deliverable in new ways, like an inhaler for lung protection against an aerosol threat.
Direct delivery to the target organ.
That's the potential. So NIH funding in these areas builds fundamental capabilities with direct defense applications.
So this really confirms it. The civilian research pipeline, funded by NIH for health and science, it's simultaneously generating the knowledge, the tools, the platforms essential for sophisticated biodefense.
The boundary is incredibly porous. Scientifically, technically,
Yet,
fighting a pandemic and preparing for an engineered bioattack draw on the exact same wellspring of knowledge and expertise.
and that porosity, that's the defining feature of your field today, isn't it?
It absolutely is. Your expertise is vital for global health, and every advance you make gets looked at through a strategic, sometimes military lens. #tag outro.
Well, this has been quite a journey, decades covered, from policy choices that, maybe unintentionally, created defense gaps, right through to today's dual-use engineering landscape. Let's quickly recap the three main funding stories we pulled from these sources.
Okay, first, that terminated U.S. offensive program 1942 to 69. Its shutdown was a diplomatic win, but ironically, it created a technical gap. It hampered early defense work by removing that high-end threat benchmark.
Right. Then second, the really alarming strategic failure around the former Soviet Union's non-proliferation efforts.
A huge mess. The Biopreparat scandal, how it hid in plain sight. The systemic oversight failures at USA, at NASA, allegedly channeling funds, including NASA space research grants, to the remnants of the Soviet BW program.
Led by a former intelligence director, no less.
Exactly. And culminating in that suppressed CDC warning. The one about U.S. funds potentially underwriting work on a mutated measles virus engineered for fast-acting AIDS-like characteristics. That whole episode just screams danger from failed strategic oversight and secrecy breakdowns.
And then third, where we are now, the current very high-stakes domestic dual-use research scene.
Right. DoD and the CIA relying heavily on offensive simulation, from planning to make modified anthrax,
Using virtual pathogens, sophisticated CFD modeling, analyzing captured bomblets.
all balanced against and critically enabled by the foundational civilian science funded by NIH. Things like rapid mRNA platforms, structure-based vaccine design, nanobody tech, research essential for health but inherently fueling advanced biodefense potential.
So let's bring it home. What's the final implication here? For you, the specialist, the professional working in the lab, in bioinformatics, in vaccine development.
When you connect all those dots, you look at the Biopreparat diversion, the NASA grants going astray under Kalinин's watch. You look at the intense internal U.S. debate over defining protective purposes under the BWC for something like DARPA's safe genes.
What emerges?
A fundamental, unavoidable tension, right at the heart of your work.
A tension between what?
Between the necessary secrecy, the strategic calculation, sometimes even deception that's required for effective state-level defense planning. Planning that needs to simulate the enemy's worst potential capabilities.
Okay, the need for defense secrecy.
And on the other hand, the urgent need for transparency, for international scientific collaboration, open data sharing, and critically verifiable compliance with treaties like the BWC in the field of microbiology.
Science thrives on openness, publication, collaboration.
But strategic defense often demands classification, proprietary knowledge, keeping capabilities hidden. You, listening, are working right in that overlap zone. The knowledge you generate saves lives. And it also represents a potential blueprint for incredibly sophisticated threats.
So the challenge for you.
The challenge is constantly ensuring that your pursuit of scientific excellence, whether it's a new vaccine design, a diagnostic tool, a gene-editing countermeasure, remains clearly and defensibly within those protective mandates of the BWC.
Asking the hard question.
Always. Does this work provide a clear defensive advantage that cannot easily be spun or misinterpreted as crossing into offensive capability development? That's the tightrope, the one you walk every single day in this field.
Thank you for joining us for this fairly intense intelligence briefing. We hope it provides valuable context.
Consider the strategic weight. Working in a field where civilian science is constantly analyzed, funded, and ultimately defined by its potential to either prevent or possibly create a global threat. It's a heavy responsibility.